Bioinformatics Micro Modules Maximum Parsimony for Phylogenetic Tree Reconstruction Part 1. In this module, we discuss the maximum parsimony method, which is a sequence-based method for phylogenetic tree reconstruction. We discuss a simple algorithm for solving the small parsimony problem and leave more in-depth discussions for the next module. The term maximum parsimony means that we want to find a tree that can match the input sequences with the smallest number of mutations. In this approach, a tree is assumed to be more likely to reflect the actual evolutionary events if it involves fewer mutations. There are two main reasons for this assumption. First, since mutations do not happen frequently, an explanation of the input sequences that involves fewer mutations is more likely than an explanation that involves more mutations. We will make this notion formal in a later module. Second, in science, we usually look for the simplest explanation for our observations, partly to avoid overfitting the observed data that could make our explanation not generalizable. There are two versions of the maximum parsimony problem. In the large parsimony problem, we are only given a set of input sequences, and we need to determine both the topology of a rooted tree for connecting the input sequences, as well as the ancestral sequences in the internal nodes, such that the total number of mutations along the branches is minimized. This is an NP-hard problem, which is a class of computational problems with no known polynomial time solution. Since this problem is so difficult, as discussed in the last module, one way to deal with it is to work on a simplified version of the problem. This leads to the second version of the problem called small parsimony. In this simplified version of the problem, the tree topology is also given, and the only task is to infer the ancestral sequences in the internal nodes such that the total number of mutations along the branches is minimized. Let's use an example to illustrate these ideas. For simplicity, for now, let's assume that each input sequence contains a single nucleotide, and later we will study the general case of having longer input sequences. In fact, by making the assumption that different sites on the sequence are independent, which is unrealistic but greatly simplifies the problem, we can study each site separately. Now, let's look at the tree in this example. It has 5 input sequences at the leaf nodes, and 4 internal nodes that represent their ancestral sequences. These ancestral sequences are the unknowns that we need to find out. In this tree, there are 4 mutations along the branches, namely the G to C mutation from the root to its left child, the C to A mutation from this left child to its own left child, and the G to A and G to T mutations on the right. Is it the most parsimonious solution, that is, the assignment of ancestral sequences, that minimizes the total number of mutations? The answer is no. Actually, for this problem, it is possible to assign the ancestral sequences such that there are only 3 mutations in total. One way to achieve it is to have 3 A's and 1 G assigned to the internal nodes as shown. Another way is similar, but with the G replaced by a T. Yet another way is to replace the T with an A. We now have 2 questions. The first question is whether these are the optimal solutions with the minimum number of mutations. The answer is yes, since all 4 types of nucleotides are present in the leaf nodes, and therefore, no matter how the ancestral sequences are assigned, there must be at least 3 mutations along the 3 branches. The second question is whether we have found all the optimal solutions. That is, whether there are other ways to assign the ancestral sequences that would lead to only 3 mutations. The answer is that there are no other optimal solutions, and we will come back to this point later. Now we want to develop an algorithm to systematically find optimal solutions. Intuitively, in order to minimize the number of mutations, we should determine the sequence of an internal node in the following way. First, if the two child nodes both prefer the same nucleotides, this node should also adopt these nucleotides. On the other hand, if the two child nodes do not have any preferred nucleotides in common, this node should keep their preferences in mind and try to adopt a nucleotide among their preferences. Exactly which nucleotide to adopt can be decided later until the parent of the current node has made its decision. These ideas actually form the basis of Fitch's algorithm, which is able to find some optimal solutions, but does not guarantee finding all of them. This algorithm works in two phases, called the upward phase and the downward phase. In the upward phase, every internal node gathers the preferences of its children to form its own preferences. For a leaf node, the preference is the nucleotide at the site of interest in the input sequence. For an internal node I, it forms a set Si of its preferences. Like what we just discussed, if the preference sets of the two children have any nucleotide in common, Si is set to these common nucleotides by taking the intersection of the two preference sets. If the preference sets of the children do not have anything in common, Si is set to be the union of them to keep all the children's preferences. This preference gathering process is performed from the leaf nodes upward until the root. The second phase then makes the decisions. The root first picks any nucleotide in its preferences as the actual choice. Then going downward, each internal node compares the actual choice of its parent with its own preference set. If the parent's choice is also one of its own preferences, this nucleotide chosen by the parent is also chosen by the current node. If the parent's choice is not within its own preferences, then the current node simply chooses one of the nucleotides in its preference set and ignores the parent. This process continues until every internal node has made the decision. Let's use our previous example to illustrate this algorithm. Starting with the input sequences and the tree topology, we have 4 internal nodes, the nucleotides of which are to be decided. In the upward phase, we fill in the preference sets one by one upward. First, for this internal node with the two children preferring A and C, there are no common nucleotides in the two preference sets, and therefore, this node keeps their union as its own preference. In the same way, the parent of the two nodes that prefer G and T also keep both nucleotides in its preference set. The parent of it now has the left child preferring G and T and the right child preferring A, and has to keep all 3 nucleotides in its own preference set, since the two children do not have any common preferences. Finally, for the root, the left child prefers A and C, while the right child prefers A, G, and T. This time, there is a common nucleotide in the two preference sets, and therefore the root takes this common nucleotide, which is an A, as its own preference. The upward phase is then completed. Now for the downward phase. As we will see, the algorithm can produce 2 solutions depending on which nucleotide is chosen along the way. First, since the root has only one nucleotide in its preference set, it has to choose it. Then for its left child, since the root has chosen A and A is also in its preference set, it has to choose A. For the right child of the root, in the same way, it has to pick A as its choice. Now, for the left child of this node, the preference set contains G and T, both of which are different from the parent's choice, which is A. In this case, it can freely choose any nucleotide in its preference set. If it chooses G, one solution is obtained. If it chooses T, another solution is obtained. We can verify that both solutions have 3 mutations in total along the branches, and are thus optimal solutions.